Welcome to zoology class. My name is Ramakrishna, senior lecturer in zoology. So today we are discussing about the IPE model paper in senior zoology as per 60 marks. So these are 60 marks totally here, 2 marks questions, 4 marks questions and 8 marks questions also. So 2 marks questions are 10, 4 marks questions are 8 and 8 marks questions are 3. In these 10 questions, no choice. We are following all answers. There is no choice. And in 8 questions in that is 4 marks. Here only 2 questions are choice. Remaining we should write 6 questions. And in 8 marks questions, 3 questions are there. In these 3, 1 question is choice. Remaining 2 questions are we should write. So today we are discussing about the one model paper as a senior zoology. So in this model paper, firstly we are start with 2 marks questions, that is very short answer question types. So in this first question is, that is what is tidal volume? Find out the tidal volume in a healthy human in an hour. So that is the question. So firstly we are discussing about the what is tidal volume. Tidal volume means, that is the a healthy human being can breathe in a normal condition and also exhalation also normal condition. So that is called tidal volume. Approximately the tidal volume is 500 milliliters. 500 milliliters in one single breath in as a normal inhalation and also normal exhalation. So it is the tidal volume approximately. As for science, one normal human being can breathe in a minute approximately 12 to 16 times per minute. So approximately values. So one normal man can breathe in a minute approximately how many milliliters of air can able to breathe. So here the approximately here 500. For example, 12 times per minute. So its volume is 6000 milliliters of air in a minute, especially. If 16 times, it is increasing. So that is the 8000 milliliters of air. So in this following, in an hour, so 16 we are calculating with the 500, that is firstly we are calculating with the 12 times and also 16 times. So finally we are calculating in an hour values. So that is the tidal value. Second question, what is autocatalysis? Give 2 examples. So it is from digestion and absorption. Autocatalysis, what is firstly Autocatalysis. Catalysis. So auto means self. Auto means self, itself. Catalysis means it is making itself. So firstly, totally here, autocatalysis means here this process is occurred in the process of digestion. Process of digestion in a protein. So autocatalysis means one enzyme that can prepare itself. So that is the definition of autocatalysis. So in the following, so for example for this autocatalysis, that is firstly the protein digestion is occurred in the intestinal actually. So firstly that is a proenzyme, that is the pepsinogen actually. That pepsinogen is that is inactive enzyme. Pepsinogen, it is a inactive enzyme. It is prepared in our intestinal glands especially. So it is activated by the firstly enterokinase enzyme. So here the inactive pepsinogen, it is converted into the active enzyme, that is a pepsin under the enterokinase enzyme. So after that this pepsinogen is can made itself, means here the pepsinogen can change into the pepsin under the pepsin enzyme. So here this active enzyme is prepared itself as another active enzyme, that is from the pepsinogen. So it is the best example for the autocatalysis. Right. The third question we are discussing, that is sino-atrial node is a pacemaker in our heart. So why? Right. Firstly, what is sino-atrial node? So it is a particular made by the nodal tissue in human heart especially. For example, so as per a nodal tissue, for example, it is the human heart. So this sino-atrial node is present at it is the right atrium, it is the left atrium, it is the right ventricle, it is the left ventricle. So the actually here the two nodal tissues are present in human heart. So these are the first one is present at the anterior corner of the right atrium, that is the sino-atrial node, and the second nodal tissue is present at that is the posterior corner of the right atrium, that is the atrioventricular node. These two types of nodal tissues are present in the human heart. So firstly, the sino-atrial node is called pacemaker. Firstly, what is pacemaker? Pacemaker means it's generating to the action potential selfly without any nerve impulse. That's why this sino-atrial node is called pacemaker firstly. So it is the location of sino-atrial node is it is present at the right anterior part of the anterior corner of the right atrium and also opening of the inferior vena cava. Opening of the inferior vena cava. So it is the sino-atrial node. So firstly it is generating to the action potential without nerve impulses. Then it is sent to the both atrias and also the signals are also received by the atrioventricular node. So finally it is also receiving to the action potential and it is sent to the action potential into the that is both ventricle by the nodal tissues especially. So these tissues are bundle of his and also it is spread as a Purkinje fibers. Purkinje fibers in a both ventricles. So that's why it is called pacemaker. Fourth question is that is a musculoskeletal system. What is triad system? It is also very important for IPE. So triad system, firstly what is triad system? Triad system means it is formed by the inversion of sarcolemma into the sarcoplasm. So for example it is the sarcomere, it is the muscle fiber. So it has a number of dark and light bands. So here these are the dark bands. These are also dark bands. So around this muscle fiber has a particular sarcolemma. It is the outer layer. Means the complete sarcoplasm is present inside of this sarcolemma. So the triad system is formed by the inversion of sarcolemma into the sarcoplasm. So finally it is look like a T-tubule. Right. So it is the sarcolemma. So the T-tubule is formed by the inversion of sarcolemma into the sarcoplasm. So finally it is here. This both side of this complete both side of this T-tubule, the sarcoplasmic reticulatum cisternas are present. This both side of the T-tubule. So the sarcoplasmic reticulatum ends are called cisternae. So these cisternae are present at the both side of the T-tubule. So that's why this system is called triad system. Okay, the fifth question is that is the neural system. So name the cranial meninges covering the human brain. So total meninges are three meninges are covering to the human brains. So these are the first one is outer meninges, second one is middle, and third one is inner. So outer covering meninges is called dura mater, and middle meninges is called arachnoid, and inner meninges is called pia mater. In these three meninges, the pia mater is directly covering to the brain, and it is very highly vascularized meninges also. And arachnoid, it is completely web like, that is web like means that is completely spider web like meninges. Between these arachnoid and pia mater, one space is present. That space is called subarachnoid space. Subarachnoid space. So actually the importance of this subarachnoid space is the cerebrospinal fluid is present in human brain only two areas in the human brain. So that is, first one is the ventricles of brain, and the second region is the subarachnoid space only. So it is the that is arachnoid, it is present between the arachnoid and pia mater. So it is the that is fifth question is the cranial meninges. Sixth one is, that is from endocrine system. What are islets of Langerthans? So these islets of Langerthans are present in that is pancreas. So the pancreas is a mixed gland actually. Islets of Langerthans. So these are the main role of these islets of Langerthans. These are secreting to the hormones especially. So firstly, actually the pancreas, these cells are present in the pancreas. Pancreas is a mixed gland actually. So it can able to produce to both types of secretions. These are the first one is enzymes, second one is hormones. So in these pancreatic cells, which cells are secreting to the hormones? So these are participate in the endocrine system. So these cells are called islets of Langerthans. These islets of Langerthans are mainly classified into two types. These are the especially main cells, main role endocrine system. So these cells are the first one is alpha cells, second one is beta cells. So the alpha cells are secreting to the particular hormone, that is the glucagon hormone, and beta cells are secreting to the insulin. So the glucagon, it is called hyperglycemic hormone. Insulin, this hormone is called hypoglycemic hormone. It means here the glucagon, when the glucose levels are decreasing in the blood, immediately this glucagon is secreted from the islets of Langerthans. Then these hormones are released into the circulation. Then these are increasing to the gluconeogenesis process. Then immediately the blood gaining to the, that is blood levels, the glucose levels are increased into the circulation, means blood. So prevention of this glucagon function, immediately the insulin also secreted from the beta cells from the, that is pancreas. So these are higher level of glucose molecules are removed from the, that is blood especially. So that's why it is called hypoglycemic hormone. If the beta cells are cannot secreting insulin, what would happen? So that is, that person may goes to the diabetes mellitus disease, that is the sugar disease especially. Why? Because that is the decreasing of insulin in the circulation, insulin in the human blood, right? That is leads to the increasing of glucose level in the blood. So that is leads to the diabetes mellitus. So it is the main function of islets of Langerthans. Seventh question. Colostrum is very much essential for the newborn baby. So colostrum. This colostrum is present in the maternal breastfeeding. It is secreted from the first few days of the maternal, that is milk. So the colostrum has a particular antibodies, that is the immunoglobulin A. So these antibodies are improving to the, that is the immune system of infant. So that's why the colostrum is very much essential for the newborn baby. Eighth one is, after ovulation, the complete graphene follicle becoming in a yellowish in color. So these yellow mass of cells are presently. What is the main function of the yellow mass of cells after ovulation? So here firstly the yellow mass is appeared in the, that is graphene follicle. Graphene follicle after ovulation. After ovulation, after completion of the ovulation, completely the graphene follicle becoming appearing in a yellowish color. So the cells are completely converted into the, that is particular color, that is the yellowish in color. So these yellow cells are particularly secreting to the particular hormone. So that hormone is called, that is progesterone. So the main function of this progesterone is, it is during the gestation period up to 4th month. It is that is maintaining to the fetus in the uterus especially. After completion of the placenta formation, these yellow mass becoming a disappear. After that, this progesterone released from the placental cells. So it is the particular yellow mass of cells that present in the follicles after ovulation in the graphene follicle. ninth one. It is the very short answer questions. That is from last unit. That is applied biology. So that is the explain the term hypophysation. So firstly here, what is hypophysation? Hypophysation means it is increasing the breeding of brood fish. So firstly, the process of hypophysation is that is stimulation of hypothalamus. Hypothalamus that is from the brain, that is part of the brain, hypothalamus. So this hypothalamus is releasing to the particularly two hormones. That is the first one is luteinizing hormone. Second one is the follicle stimulating hormone. These both hormones are actually these are the neural hormones. They are also called neural hormones. Means here the hypothalamus that is these are from the released from the pituitary gland. The pituitary gland part of the that is the adenohypophysis region. Adenohypophysis part. It is releasing to the adenohypophysis part of the pituitary gland. So these secretions are secreted from the adenohypophysis part of the pituitary gland. These secretions are initiated to the that is gonads especially. So finally these hormones are stimulated to the gonads, and immediately that brood pouch is releasing to the particular breeding season. So the main process of this hypophysation is that is improving the breeding of brood fish. It is the hypophysation. Define the transgenic animals. So mainly transgenic animals, definition of these transgenic animals means that is generally animals had their genome and also had their DNA. So it is manipulated. It is manipulated to possess extra express characters is called that is the transgenic animals. So the best example for the transgenic animals is that is completely approximately 95% of animals are the mice. These are the best example for the transgenic animals. So mainly, so the main uses of this manipulation of this genome means that is the DNA nitrogen bases also. So the main uses are, it is mainly used for the, it is preventing to the genetical disorders. It is mainly preventing to the genetical disorders, and also the manipulation of physical and biologically. So number of uses for the uses about these transgenic animals. It is the transgenic animals and its definition.